Hey everyone! So welcome to the online video lecture covering Chapter 17. This chapter covers translation and transcription, but more in detail than Chapter 16 did. Now remember, the central dogma essentially states how do we go from DNA to proteins. So it goes DNA, RNA, proteins. DNA to RNA is transcription, RNA to protein is translation. So now what we're going to talk about, now that we've kind of discussed the mechanics of what's needed for transcription, we're going to talk about the actual process of transcription. Now it occurs in 3 steps. First, initiation. Second, elongation. And then third, termination. So 3 fairly simple steps. You need to start it, you need to make that mRNA really long, and then you need to stop at some point. So let's talk about initiation first. So the very first step is we need to get RNA polymerase to that strand of DNA to be transcribed. Now, depending on eukaryotes or prokaryotes, it works a little bit differently. This is also where you should start noting the differences on that sheet I gave you in class between transcription and translation in eukaryotes and prokaryotes. If you need another copy, I have put it on Blackboard for you to print your own copy. So initiation, the very first step. The sigma factor, or those basal transcription factors, depending on the species, is going to bind to that promoter. Again, the promoter, it's not coding for a protein. It's on the non-template strand, and really it just serves as a starting place. It tells RNA polymerase, hey, this is where you're supposed to begin. So the sigma, or the BTF part of that holoenzyme, its whole job is just to go through that DNA molecule and bind onto promoters. Now, once sigma factor, once the BTFs bind to the promoters, that's going to attract an RNA polymerase to it. Now, you don't really see that in these images. You kind of see them as one unit. There's a video and a couple of slides that'll show this process a little better, but they are separated. First is the sigma factor, and then RNA polymerase will come and bind to that. So you can see in this second image that this DNA molecule, the double-stranded molecule, is being weaved through this RNA polymerase, or RNA pol, just for short here on the slide. So what you see is DNA starting to unwind. Now, it's not the entire strand. It's really just the part where we're starting to replicate. Here's this promoter region right here. So there's the promoter, and then here we have ribonucleotides being added to this DNA. This is not a primer. Again, RNA polymerase can just start adding no matter what. So this DNA is being opened. And this active site, which you see down here, this active site is where ribonucleotides are actively being put on and copying this DNA strand. And remember, it's copying the template strand because RNA polymerase can only move 5' to 3'. So as you see in this image, RNA synthesis has already begun. Right now we have, it looks like 4 ribonucleotides attached. So the promoter told us where to start. You see right at the end of this blue section of the promoter, we've already started putting on ribonucleotides. So it's going to continue going down. And again, this is all just initiation. Where do we start? Well, we start right after the promoter. The promoter just tells us where do we latch on to. Now finally, at the end of initiation, the whole point of the sigma factor and BTF was just to get it started, to say, hey, here's where you're supposed to start, and that's it. Well, RNA polymerase has already started. So you no longer need that sigma factor or those basal transcription factors to hang around. So they actually leave because they don't really serve a purpose anymore since RNA polymerase has already began. So that was all initiation. We started with sigma, bonding to the promoter, RNA polymerase bonding to that, and then kind of situating that DNA so that we could unwind it a little bit and add those ribonucleotides. And now the sigma or the BTS have left. So the second step, we've now started it. Now we want to elongate it. We want to make our entire mRNA molecule. And again, we're talking about transcription, and we're talking about trying to get from DNA to proteins. So here's a zoomed in image of what's happening in RNA polymerase. So here you see this mRNA, and you see it's already starting to get much longer than it was. You can also see this is the old DNA that's already been copied, and there's the remnants of that promoter. So this RNA polymerase begins moving downstream, as in it was upstream, and then it just keeps moving downstream. It just keeps moving down this DNA molecule to copy it. As I've mentioned before, RNA polymerase, its whole job is to unwind the DNA, and it's going to make this mRNA transcript from the 5' to 3' direction. And you see that down here. Here's mRNA, the 5' end. Remember, 5' means that's where the phosphate is. So you follow this down. Here it is right here. So where my arrow is, that's the 3' end. That's where the hydroxyl group is, and that's what's necessary for RNA polymerase to add new nucleotides. Same thing as DNA polymerase. You see two labels on this image. There's the rudder and the zipper. And they actually kind of mean the same thing that they mean in our cultural societal complex. So the rudder, you see how it's kind of it's orienting this template strand. It's kind of pushing this template strand down and kind of pushing it towards this active site. Because remember, the active site are where ribonucleotides are coming in. So the rudder on a boat is used for directions, for steering left and right. Well, it's the same thing here in RNA polymerase. Its whole goal is to direct or move that template strand to where it needs to go, which in this case is towards the active site. Then we also have the zipper. Zippers are kind of meant for, bringing it together or putting it apart, depending if you're zipping up or zipping down. Kind of similar here in RNA polymerase. In RNA polymerase, the zipper, its whole goal is to keep these two molecules, mRNA that's already been created, already been transcribed, and this DNA separated. You want to be pushing this mRNA out. You don't want it to stay stuck in that DNA template. So the whole point of the zipper, as you can see here, here is RNA being duplicated, being added. And it goes up, but then it hits this zipper, which kind of pushes it downward. And it's pushing it downward out of this enzyme, and then eventually it'll be released and out into the cytoplasm. So rudder is just going to help direct that template to the active site. The zipper is going to help direct mRNA away from DNA. We want to keep these molecules separated. And then finally, as you can see in this image, DNA doesn't stay split. DNA does come back together. So if you look here, so this is the area that was just transcribed. Here's the new area to be transcribed. So here's everything that we've already made a copy of. And you notice it's already back in its double helix. Think back to DNA synthesis real quick. Remember, we had those single-stranding binding proteins. Their whole job was just to prevent that DNA that was being unwound from coming back together. Well, here we don't have those SSBPs. So that's how you see this double helix reforming all on its own. It doesn't take enzymes to do it because DNA wants to be in that form. And finally, we have termination. So we've copied all of that gene from that DNA, and that's it. We don't need to copy anymore because we've already coded for all the amino acids we'll need for that protein. So with termination, it begins when RNA polymerase copies a code off of the DNA. DNA had a promoter area. It also has a termination area. Now, the termination code kind of varies quite a bit between individuals as prokaryotes, individuals as eukaryotes, and between both of them. But we'll keep it general and just say it's going to code some sort of terminator sequence. Now, it does work differently between bacteria and eukaryotes. In bacteria, this final termination, so that's what I'm circling right here, this final termination signal is just a whole bunch of various letters, but when RNA is making a copy of that, that specific sequence of letters, whatever they are, actually makes mRNA form this hairpin structure, and that's what you see in this image, this nice hairpin. And again, that's formed just because of the certain type of base pairs that were in this termination signal. And you see that in a lot of different bacteria, that whatever combination that is, it actually perpetrates the creation of this hairpin. Now, this hairpin, it kind of takes up space, it kind of pulls the mRNA. That will actually help it to pull away from RNA polymerase. Eukaryotes work a little bit differently. They too have a termination sequence, but it's a series of A's. So it's going to be, oh, here's for this gene, here's for this gene, here's for this amino acid, here's for this amino acid, A. And then it's just going to keep coding random things. Well, that segment of A's will actually kind of send a signal out to the cell saying, hey, this protein or this mRNA has finished. It has all of those A's on it. It must be ready to be cut away. Because right now, RNA polymerase is still going. RNA polymerase is still copying things, but we don't want it to copy things anymore. So an enzyme recognizes that A sequence and will go and cut that mRNA. So it'll cut it, say, right here. It'll cut that mRNA and say, okay, this is it. I see this poly A signal that's telling me it's time to cut it. So for either of those, whether it's the hairpin structure or it gets cut by an enzyme, one way or another, that RNA transcript, that molecule of mRNA, gets released. The second thing is that once you get rid of mRNA, well, RNA polymerase doesn't have to be there anymore. Now the sigma factor and the BTF has already left, and now RNA polymerase is going to leave because it doesn't need to be around any longer. So this is just an overview slide of transcription in both prokaryotes, which is on the left, and we know this is prokaryotes because we see sigma. We see this -35 and -10 box. And on the right-hand side are eukaryotes. We know that because we see this TATA box. There's also transcription factors, or the basal transcription factors. So that's the end of this first video. What I recommend doing before you begin the next one, take a look at this video. It provides a really great overview of transcription. And then also fill in that data table with the differences between eukaryotes and prokaryotes when it comes to transcription. When you're ready, go ahead and start watching the second video.